And now for our last section in our Chapter 5 video lecture series, Section 5.6 on tissue development and aging. Here are your learning objectives. And we'll start off with tissue development. First, we're going to have our oocyte, or the egg from the female, become fertilized by a sperm cell. That is going to form a diploid cell, diploid meaning we have 46 chromosomes here now, and we're going to call this a zygote. And that zygote begins to multiply. So from one cell we get 2 cells, from 2 cells we get 4, from 4 cells it keeps on growing. And after multiple cell divisions, we get our blastocyst. And our blastocyst has cells that are starting to form, and we are going to implant on the uterine wall. And as the cells continue to form, we are going to have our embryoblasts that are going to create our embryo. And this is an image of an embryo down over here. And as we are forming the embryo, we see that there are 3 primary germ layers that are formed by the 3rd week. And that is our ectoderm, our mesoderm, and endoderm. Now let's go ahead and talk about each of these primary germ cells, but I also want to draw your attention to these little squares down here that give you an idea of what these primary germ layers are responsible for forming as the embryo grows. Now for our ectoderm. This is initially located on the dorsum and external embryo surfaces. So you could see that over here as it's developing, that they're going to be responsible for forming a lot of external structures. You can think ecto is for external. So we're going to see this create the epidermis of our skin, hair, and nails, and our exocrine glands. And some but not all epithelial tissues are going to be derived from this ectoderm. And other structures that it is derived from will be the tooth enamel, our eye lens, adrenal medulla, and all of our nervous tissue. So that's what you're seeing in this illustration over here, is focusing on this top portion down over here. And you can see how this neural plate is turning into a neural tube. And this neural tube will eventually develop into the spinal cord and the brain. Now onto our mesoderm. This is going to be our middle primary germ layer, and it's going to form all of our muscle tissue. It forms the epithelial lining of our vessels and serous membranes and becomes mesenchyme. So later it's going to form all of the connective tissue within our body as well. So remember, our bones, our cartilage, all are types of connective tissue. And other mesoderm-derived structures would be the dermis of our skin, our adrenal cortex that we find on top of the kidneys, our heart, our spleen, which you find over here on the left, right under the rib cage, our kidneys themselves, the ureters, and our internal reproductive structures. And lastly, we have our endoderm, which is the innermost germ layer. And the structures that it creates are the epithelial linings of the tympanic cavity and auditory tube, our epithelial linings of the digestive tract, our respiratory tract, and reproductive and urinary tracts. Also the thyroid gland that we find in the neck, and the parathyroid glands that we find posterior to our thyroid gland, and the thymus that lies right on the heart. We'll also see that it forms portions of the palatine tonsils and most of the liver and gallbladder and pancreas, those accessory organs of the digestive system. Now we need to talk about some tissue modifications that take place. So I wanted to give you an idea of what physically happens to these cells. So here is our normal set of cells. When our cells go through hypertrophy, that means that they are going to increase in actual size. So you probably have heard of this term when it comes to increasing your muscles or muscle size. So we're increasing the actual muscle cell itself. In contrast, hyperplasia would be when we increase the number of cells within that tissue. So hyperplasia is not really possible in muscles, at least through the research that has taken place thus far. Now, if you don't exercise those muscles, you go through something called atrophy, where we're going to see a decrease in the size of those muscle cells. And this could be due to normal aging or, like I mentioned, disuse of the actual cell. Examples would be being bedridden and the skeletal muscle fibers become smaller, though it is reversible using physical therapy and exercise. And here's a clinical view. We're going to talk a little bit about stem cells. Our embryonic stem cells are going to originate from the dividing cells of that zygote that we produce, or the embryoblast that can be found in the blastocyst. And they may be totipotent or pluripotent. So the totipotent stem cells are going to be the cells that are formed from the zygote and give rise to every differentiated cell in the organism and the placenta itself. And then our pluripotent stem cells are going to be derived from this embryoblast portion of our blastocyst. This type of stem cell has capability to mature and specialize into a cell within any tissue in the body except for the placenta. Now, when we look at adult stem cells, they are undifferentiated stem cells in the body after birth. So they may be multipotent or unipotent. Our multipotent are going to be stem cells within our red bone marrow, which we find over here, and are capable of differentiating into a restricted number of some cell types, but not others. And unipotent stem cells, such as our epithelial stem cells, are going to have the ability to differentiate into a single cell type. And continuing with our tissue modification, we can have metaplasia take place. This is when we have a change of our mature epithelium to a different form. So it can occur as the epithelium adapts to a certain environment. So the best example of this is within smokers. They're going to see these metaplastic changes take place in their trachea epithelium. We have at first our normal pseudostratified ciliated columnar epithelium, which remember we called respiratory epithelium, and it becomes non-keratinized stratified squamous epithelium, and can revert back to this if a person quits. But the reason that they are changing into this tissue type, and you could see this change taking place over here, is because the stratified squamous epithelium might be better able to handle the harsh toxins and chemicals that are found within smoke and tobacco products. Now for dysplasia. This is when we have abnormal tissue development. So again, here are normal cells, and you can see that in dysplasia, we just have this disorderly growth that is taking place. We'll see that these cells will vary in shape and size, and there is evidence that the cell-to-cell interaction has broken down as well, resulting in a disorganized appearance of the tissue. And it may be precancerous, or it can revert back to normal, but it must be closely monitored by professionals so that we can start treatment in case it does turn cancerous. An example of this is cervical dysplasia due to exposure to the human papillomavirus. So over here we have our normal cervical cells that a woman getting a pap smear would experience. So we would see this on a slide. And then here is someone who has a mild dysplasia, and you could see that we have a slight changing of those cells. Maybe their nucleus has increased in size. The cell itself has also increased in size. Sometimes we can see multiple nuclei within one cell. And I like this little scale here because it shows you kind of the change that we would have take place in order to be in different diagnoses. Now for neoplasia. This is when we have tissue growth that is out of control. And we did talk about a few different types of neoplasia already. So we'd say all of these are neoplastic. And this is when we can have a tumor or abnormal tissue develop. And they could be either benign or malignant. When it is benign, that means it is localized to one area for growth and it does not spread. Whereas if it is malignant, it metastasizes or spreads to other tissues and starts to invade those tissues. And this is cancer. It can interfere with the normal functioning of that tissue and lead to death. Now for necrosis. This is tissue death, and it is irreversible. It is our inflammatory response to any type of tissue damage, and we are going to discuss gangrene as an example of this. So our necrosis of soft tissues within our body is what is happening in gangrene, and it's due to a lack of arterial blood supply, meaning this tissue is not receiving oxygen. And here are some of the different stages of gangrene. We have stage 1 all the way to stage 5, and here are our descriptions. It is going to be common in our limbs, especially in our fingers and toes, and is a major complication of diabetes since we don't get enough blood supply to these areas. We can also have an intestinal gangrene, in which we are going to see an obstruction of blood supply to the intestines, and so our intestinal tissue is going to start to die, and this can lead to death. And here there are different types of gangrene. We have dry gangrene, which is pictured over here. This is involved part in desiccated, meaning dried out and shriveled tissue. It's usually due to extreme cold. So you might know this as frostbite. Then we have wet gangrene, and this is caused by bacterial infections of our tissues with a loss of our blood supply. And we've got ruptured drying cells that are going to release fluid, and that fluid is why this is called the wet gangrene. And this allows bacteria to flourish even more. And then we have our gas gangrene, in which bacteria are invading necrotic tissue, and bacteria produce gas bubbles, in which we're seeing these almost blister type look. And next we're going to discuss aging of tissues. All of our tissues are going to change as we age. We want to get proper nutrition, good health, normal circulation, and of course not have any wounds take place in order to have normal functioning. And support, maintenance, replacement of our cells, and that extracellular matrix should all be taken care of in order to get that normal functioning as well. But after we hit our middle age, we start to see that these structures or these functions become less efficient. And the structure and chemical composition of a lot of our tissues become altered. We see that our epithelia thins out, our connective tissue starts to lose its pliability and resiliency, and collagen that's also in relationship with the connective tissue is going to decline. And these top 3 things is kind of what results as wrinkles. And then our bones are going to become more brittle, leading to fractures, and our muscles atrophy. And that is it. Let me know if you have any questions at all on Chapter 5.